Thanks, Grant. Hi, everyone. Thanks for turning up. Thanks, Grant, and also Cyril for facilitating today and organising for me to come over. I'm really grateful to both of you for organising this. So what I'm going to talk to you about today, as Grant said, is essentially what I've been working on since about 2013. The bulk of this work has been done over at Sunshine Hospital in Melbourne and also in collaboration with some researchers that I've been working with at Harvard University in the US. So I'm going to be talking about some cell-based molecular work, but also I was part of a worldwide competition in 2019 run by Oxford University, and I represented Melbourne Uni, and I got flown over to Oxford to talk about this stuff at the global final, so I'm going to combine some of that in with the molecular work as well. So, oh, my clicker isn't working. And I'm just going to have to keep going back and forth to the computer. Computer's frozen as well, which is not great. Thanks. So what I'm going to be covering is firstly a little bit of background on breast cancer, just to bring you all up to speed. I'm going to talk then about the big picture, talking about stress and how it has an interaction with breast cancer. The bulk of this talk is going to be molecular biology. Sorry to some of you that that's of no interest to, but that's the main majority of my research findings. And then I'm going to do a summary of all of that at the end and where I'm hoping to take the research next after that. So breast cancer is the second most commonly diagnosed cancer in Australia after melanoma now. Unfortunately, 1 in 7 Australian women will be diagnosed with breast cancer in their lifetime, which is a really unfortunate statistic. It's a heterogeneous disease, so not everyone's cancer is alike. There's lots of different subtypes. I'm going to be focusing on 3 in this talk today. So luminal, which is a more breast-like tumour. HER2 breast cancers, which are human epidermal growth factor receptor 2. This is a specific mutation that triggers cells to grow and divide. And the third type that I'll be referencing briefly is basal or triple-negative breast cancers, and these are quite nasty or aggressive. They don't have a very good survival rate. The bulk of the research that I'm going to be focusing on and talking to you about is on this second type, which is HER2 breast cancer. And the reason that I've done a lot of research on this and why I'm focused on this and passionate about this is, like Grant referenced in the intro, because of my mum. She was diagnosed with HER2 breast cancer during my PhD. She was given special targeted treatments that target that HER2 receptor. And unfortunately for my mum and everyone close to her, the treatments didn't work and the cancer progressed and it metastasized to her brain and she died as a result. So metastasis is the unfortunate outcome if cancer treatment doesn't work. And there's a growing body of evidence that shows that stress plays a role in this process of metastasis. So stress can promote most of the hallmarks of cancer, things like survival, allowing a cancer cell to grow and divide in an area of the body that it normally shouldn't be. It can allow cells to migrate and also invade other parts of the body where they shouldn't be. So you can see this is a picture of some cells that I'll talk to you a bit more about later in the talk. But these cells are being placed under stress. They're breast cancer cells. They're proliferating, migrating, and invading other areas of this 3D matrix that we have them growing in. So there's research that shows that stress plays a role in this function of metastasis. From impaired immune function, we know that the immune system is really important for suppressing developing cancer cells. It can locate cancer cells, neoplastic cells, and eliminate them from the body. Stress depresses that process. There's also lots of animal models that we have now of psychological stress, restraint stress, light stress, social, I've forgotten the word, where the animal gets stressed by another mouse in its vicinity. Social confrontation, that's the word I was looking for. Social confrontation stress, all of these things, all of these different types of psychological stress in animal models have been shown to be associated with these markers of cancer progression. What I'm going to be focused on, and I'll talk about this a bit more later on, is cellular stress. So at the microscopic or molecular level, stress can also play a role within the molecular biology of the cell to promote these different markers that allow a cell to migrate and metastasize. So the two main systemic ways that we know that stress plays a role in cancer metastasis and progression is through adrenaline and glucocorticoid pathways. So you might be familiar with this. I've got a little picture over on the left. If someone, an animal, a person undergoes some kind of stressful event, that may be even something like cancer diagnosis, their adrenal glands will release adrenaline, and that adrenaline, that hormone circulating in their system, will bind to adrenergic receptors. We've got a fair amount of evidence now that shows that activating adrenergic receptors is associated with things like migration, invasion, and even therapy resistance in an in vitro setting. Interestingly, beta blockers, which are a type of drug which bind to these adrenergic receptors, block them and inhibit the adrenaline from binding onto the adrenergic receptors, have been shown to have an association with decreased recurrence with metastasis in breast cancer. And there's lots of clinical trials being undertaken right now, as we're talking, using beta blockers. There's one actually going on at Monash University in Melbourne, using beta blockers as an adjuvant therapy within treating breast cancer to try and limit this process of breast cancer recurrence. The second main physiological pathway we know of is with glucocorticoids. So look at this little diagram over here on the left. We've got a stressful event. Again, this different stress hormone is a glucocorticoid, cortisol, which is a slow-acting stress hormone with a longer half-life than adrenaline. And cortisol will bind to glucocorticoid receptors in your body. Now, there's interesting research, some of which only came out a couple of years ago from the Swiss group, that shows that patients with high tumour expression of glucocorticoid receptors have worse survival. And secondly, and really importantly for clinical practice, glucocorticoid agonists promote metastasis and decrease survival in animal models, mouse models. And why this is such a big finding, only came out in late 2019 in Nature, is glucocorticoid agonists, things like dexamethasone. These are drugs that are routinely, fairly routinely, given to cancer patients to help them deal with the negative side effects of chemotherapy and other things. So cancer patients are often given these drugs, and now they've found that these drugs might actually be promoting metastasis. So not a great outcome. This is the two main physiological pathways that can promote metastasis, the two main pathways of stress. But there are lots and lots of different stress points around a woman when she is diagnosed with breast cancer. And two of the ones that I want to talk to you about just quickly are firstly social support. So there's evidence showing that social support is really important for a woman with breast cancer. There's evidence that women who feel isolated, depressed, lonely, have a much poorer prognosis for their disease and for treatment as well. This is particularly difficult, particularly hard for women from rural and remote areas because they often have to travel a long way for treatment, as well as First Nations women who have to obviously come to the city or somewhere, leaving their family, their support networks. So this is an additional confounding variable in the whole treatment pathway and could potentially be affecting the outcome of their disease. Another second main big factor for women when they get diagnosed with breast cancer is the financial stress, or that playing a role within the treatment and the outcome of their cancer. There's a number of different aspects that come into play here with the financial stress. Firstly, a woman might have to reduce her hours of work. She might have to stop work completely because of the treatment, the effects it has on her body, having to go to the hospital for continually getting treatment, even the small things, day-to-day things like paying for additional transport to and from the hospital every day, paying for parking at the hospital. These additional financial stresses could be playing a role, potentially, in cancer, knowing that adrenaline, cortisol, these stress hormones can play a role with metastasis. Another key thing, another key factor with the financial stress is the actual cost of the medications themselves. So I should have made that figure before, but I forgot to do that. Anyway, we'll skip to the next slide. So the cost of treatment without government subsidies here in Australia can be prohibitive for most patients. So if we're thinking about one of the key drugs which is given to women with HER2 positive breast cancer, like my mum, she got Herceptin. That costs approximately $50,000 a year in Australia. We're pretty lucky, we have a pharmaceutical benefit scheme here that will bear most of that cost. When my mum was diagnosed, nearly 20 years ago now, for the first 2 years in Australia, if you wanted to access this drug, you had to pay for it upfront. That's a lot of money and it's prohibitive for most people. In places like the US, you'd be looking at double that cost. It can be up to $100,000 a year, which is why medical costs currently in the US are the #1 cause of bankruptcy over there, unfortunately. Anyway, in Australia, if you wanted to access this drug, the first 2 years before it was on the PBS, you would either have to, from case reports I've read, tip into your superannuation, remortgage your house, sell your house, something like that, or just not access this potentially life-saving treatment. So this financial stress is an added burden into an already difficult time for a woman. As I spoke about before with my mum, these treatments, even though they're targeted and they're great drugs, they don't work for everyone. Everyone's cancer is slightly different. And so because of that, we need more research to improve the personalisation of these cancer drugs. Because if we can personalise, if we know a drug is going to work, then we can firstly potentially save some money from the government perspective, subsidising a drug, a very expensive drug that isn't going to work for the patient, and B, more effectively treat that patient by giving them a drug that's going to be more effective at treating their disease. Know that early diagnosis and personalised treatment leads to better treatment outcomes. I'm just going to pause there and have a drink. Okay, so this is where I'm going to take you back and talk about the focus of my research. This is where it's going to go into more of the molecular biology stuff. So with my research, I'm going to focus on stress at the microscopic level, cellular stress and cancer. So if we're thinking about stress to the individual cells, it can be triggered by lots of different things. Heat, chemicals, infections, all of these different stresses on a cell will trigger something called a heat shock response. And this is an evolutionarily conserved process present across all multicellular organisms and acts to protect the intracellular proteins, prevents the cell from undergoing cell death like apoptosis. By their very nature, cancer cells are stressed cells. They have genetic mutations, they have aberrant processes going on. They're often living in places in the body where they shouldn't be. They don't have access to enough oxygen or nutrients that a normal cell would be. So they're being placed under a lot of stress. And what the research shows us is as the cancer becomes more aggressive, potentially metastasizing and spreading to places where it shouldn't be, its requirement for heat shock proteins, or its reliance on them, increases. So with increased heat shock expression in the cancer cell, it's associated with a reduced therapy response and also a reduction in overall survival. The key switch for this, the on-off switch if you like, is heat shock factor 1, so the master regulator, which means if we trigger heat shock factor 1, if we switch that on, essentially, it's going to cause heat shock protein expression within the cell. So we get heat shock factor 1, we get a stress response in the cell as a result. And just to link you back to what I was talking to you about at the start with the two main physiological pathways, there's research that shows that if you have excess glucocorticoid receptor expression in the tumour, that will trigger heat shock factor activation. So we've got an interaction between what's happening systemically and what's happening microscopically within the cell itself. So here we go with the molecular biology. So all of this work, starting back way back 10 years ago now, was sort of sprouted from the idea that came from the main researcher that I collaborated with on all of this work. So this comes from Chao Min's PhD thesis. And what Chao did as part of her thesis is she took some normal breast cells and she genetically manipulate them to make them oncogenic. So when I've transformed here, that's what I'm talking about. Taking normal breast cells and make them oncogenic by adding an oncogene called RAS. So what she's done here is a migration assay. So we've got two chambers in an in vitro setup. We've added some chemoattractants, polyphenol albumin on the left, epidermal growth factor on the right. We've put some cells in the left chamber, we've got some chemoattractants in the right chamber, and we watch the cells migrate across. And these little purple dots are the cells that have moved or migrated. You can see what happens in the control. We've got a graph of the actual cells counted over here. They do move towards the chemoattractant, but if we genetically modify those cells to stress them by adding HSF1, they move much more. So this is illustrated here in the graph on the right-hand side. We've got significantly more movement or migration when we add stress to breast cancer cells that have been oncogenically transformed. So that was Xiao's finding, and then the start of our research collaboration, what I did is I tried to replicate this with some HER2 breast cancer cells, and we got the exact same result. So these are HER2 breast cancer cells. The designation is just SKBR3, that's what they're called. We've got some control, just standard breast cancer cells, and then these are different genetic modifications to make them more stressed, to add stress to the cellular environment. And what's happening, you can hopefully see here with the cell number, is more cells are moving across, they're more migratory when we add stress into that intracellular environment. So that's true for normal cells that are oncogenically transformed, and we also found that when we replicated that process with the HER2 breast cancer cells as well. The next thing we did was to look at the growth rate. What happens to the growth of the cells when we add stress in the environment? In the green, all of these different lines of different cell populations, these are control, just normal, regular HER2 breast cancer cells. They grow quite quickly, but when we modify them, genetically manipulate them to switch on HSF1 or to make them more stressed, it actually slows the growth rate down. And this is a significant difference over time. So the normal breast cancer cells are growing faster, and the stressed breast cancer cells are growing a bit slower. And I'll talk a bit more about this at the end, but we think this is a really interesting finding. The next thing we did was what's called a microarray. So we did a comparison between some normal breast cells here on the left that were unchanged. So these are the control cells. These are a black and white image. This is different stains for the same images. All of these panels on the left-hand side, these are normal breast cells. The immortalized cells are quite similar to what you find in the normal breast. Control cells and ones that have been genetically manipulated to have stress added to them. What we've got on the right is the oncogenically transformed breast cancer cells. So they've had a RAS gene, an oncogene added to them. They're more similar to breast cancer. And you can see what happens, the difference in these panels compared to the ACNI, the little spherical structures on the left-hand side. Their growth with stress is much more disorganized, much more of a metastatic phenotype. So it's able to spread and to grow into areas of the body that it normally wouldn't be able to inhabit. And this difference between the normal and the transformed cells, this more disorganized growth pattern, was significant when we measured it. Now, we did a microarray screen on these different cell populations, which is essentially looking at a comparison between the genes that are expressed in the normal cells with stress, without stress, and compare that to the genes differentially expressed in the transformed cells without stress and with stress. And we came up with one key factor, one main hit, and that hit was EGR4, which is epidermal growth, early growth response 4. Lots of acronyms in this talk. So EGR4, we came up with that because what our analysis showed is that it's been switched up and switched down in these different cellular contexts. And we did a Western blot, which you can see a little bit of behind the screen over here on the right side. So this is a Western blot showing the protein expression, relative protein expression. So with stress in the normal context, we get more EGR4. And with stress in our oncogenically transformed context, it's being switched down. So different things are happening to it in each of these different scenarios. So we add stress to the normal and it goes up. We add stress to the cancer cells, it goes down. We looked at this protein then across a whole panel of different breast cancers. I've included the pathway for HER2 over here on the left, just to help with the conceptualization a little bit. Now what we observed when we looked at all these different breast cancer cell lines, you can see some of them are HER2 breast cancers here in the middle, some of them are from luminal breast cancers, some of them are from these more aggressive basal breast cancers. What we saw is, you can hopefully see it as well, if we have activation of these receptors, either HER2 or HER1 is also confusingly called EGFR. So if we have either activation of one of those two, here or here, we have expression or a stronger expression of the EGR4 gene, which is a transcription factor expressed in the nucleus. So triggering the start of the pathway is going to end up with more EGR4. And the other thing you might be able to notice from this plot is we've got this inverse relationship between EGR4 and HSF1. So we've got this interaction happening that we noticed on the previous slide with the context of adding stress or not. We've got this across the whole suite of breast cancer cell types here. So when we've got more EGR4, we have less HSF1 and then vice versa in some of the other cells. What's interesting, at least because we were working on it, is when we were doing these Western blots, the bands should have been a certain size. We were expecting them, and what the antibody told us on the packet when it arrived was it's supposed to be around 60 kilodaltons. And none of our results were showing that. All of the bands, all of the blots that we were doing were showing a much smaller protein. We tested the protein, we tested the antibody to make sure it was picking up the right thing, and yeah, we were getting it in positive controls and not in the negative controls, but we were still getting this much smaller protein than what we expected. So we thought there might be something going on because we tried to actually clone or sequence this gene from DNA that we'd extracted from these cells, and we couldn't do it using primers on the either end of the gene. So that led us to think maybe there's some kind of splicing being involved. So the full length EGR4 gene is shown at the top here. It's made up of two exons, a smaller exon 1 and a much bigger exon 2. We looked at the whole sequence, had a look at the start sequences, I should say, within the gene sequence, and we came up with 3 possible transcripts. Transcript 1, which is probably made up of 2 exons, transcript 2, which is also being initiated in exon 1, and then this third possible transcript, which we deemed transcript S because it's shorter, and also because we think it's the one that's been regulated by stress. So S for short, splice variant, and S for stress. And we think it's being translated solely from exon 2. We weren't able to clone this gene using any primers in exon 1. We could only detect it within exon 2. To sort of support our hypothesis, we did some further experiments on this. One of which was run by an informatician, his name is Martin, who's very good over at Harvard, and he looked at a whole panel, 56 different breast cancer cell lines, and looked at the relative expression of exon 2 versus exon 1 within these different breast cancer cells. So you can see from our 56 different breast cancer cell lines we've got here, 41 of them expressed exon 2. 41 of them had exon 2 expression. In the 13 that had both, you can see that the exon 2, which is yellow, is expressed much more strongly than exon 1. This is a log scale, which means that this is a factor of, it'll be a factor of 8, that'll be a factor of 16, and so on and so forth. So there's much greater expression of exon 2 relative to exon 1. In many of them, 28 of them, it's only exon 2 that's being expressed. So that kind of supports our hypothesis for this transcript that's being spliced out solely from exon 2 rather than something that's being made up of both components, both exons of the gene. Another analysis that we did was on a US-based population, looking at over 1,000 different patients, so 1085 patients, so it's a pretty big sample size. And this is showing you, again, the expression level of exon 2 versus exon 1 in the tumours taken from all of these different patients. So the black line in the middle is the median expression. Again, this is a log graph, so we've got exponential levels of higher expression. And exon 2 in all the different subtypes that we analysed is expressed much more strongly than exon 1. And of note, this fourth band just here is the HER2. So the median expression of exon 1 was 0 in HER2 breast cancers, and is much more strongly expressed in, sorry, I misspoke there. The exon 2 is solely expressed in the HER2 breast cancers. We also looked at EGFR expression in breast cancer patient biopsies. So these patients are taken from hospitals around Victoria. We looked at 30 different patients. Again, we ran Western blots. These dark bands are showing you the protein expression. So we've got each patient, we have a normal breast sample and a matched tumour. So we're looking at two different samples from the same patient. And you can see that there's a bigger band in a lot of the normal tissue for some patients. This smaller EGFRS band is only detectable in the tumour and not in any of the normal samples that we looked at. So we looked at 20 different HER2 patients, and in 80% of those, they had this smaller EGFRS band. None of it was found in the tumour, sorry, in the normal tissue. So we think that this EGFRS is solely expressed in the tumour and not in the normal tissue, which is interesting for us at least. We then did a little bit more work directly genetically manipulating the stress levels within these different breast cancer cell lines. Remember that HSF1 is the master control switch for the stress response within the cell. So when we add stress to these different cell types, we've got this inverse response with EGFRS. So you can see that it's strongly expressed in the control cells. When we switch on stress, it goes down. That's true for the MCF7, T47D, which are luminal subtypes. It's true for the HER2 examples. And then we did the inverse with the basal cells, which are quite aggressive cells and by their very nature are more stressed cells. So what we did here, the control cells are quite stressed. We did a genetic modification to switch off HSF1 and reduce the stress. And you can see the reverse has happened in these cells. We've got low levels of EGFRS in the control that are stressed, and then more EGFRS when we remove that stress. So we've got both directions happening. We also did some drug inhibition studies. So these two drugs, erlotinib, lapatinib, they're used clinically for people for women with HER2 positive breast cancer. Erlotinib binds to one of these receptors, specifically the EGFR or the HER1. Lapatinib binds to both. So lapatinib can bind to the HER2, it can also bind to the HER1. We're going to get a stronger response as a result, a stronger inhibition using lapatinib versus erlotinib. And that's what you can see here in the Western blots I've got on the right-hand side of the screen. So as we increase the dose of lapatinib, it's binding both components of this receptor. You can see that internal components of this signaling pathway that AKT reduces, and so too does the EGFR, the downstream component of that. Erlotinib, we did the same thing, remembering that erlotinib only binds to one aspect of this heterodimer receptor complex. We get a bit of a reduction in the intracellular signaling components and a reduction, but not as apparent within the EGFRS, the downstream component. We're also able to observe that within patient biopsies that we looked at. So again, normal tissue, tumour tissue taken from the same patient. In patients that received pre-biopsy treatment, so they were given some kind of targeted therapy, could have been alotinib or lapatinib. If they were given that drug before we took their biopsy, we saw no expression of the EGFRS, as opposed to others where we have HER2 and we got the EGFRS expression within their tumour. So we're seeing, not too far. We're seeing this same pattern of expression replicated in, going all over the place, replicated in the tumour samples taken from the patients. Okay, so this study that we ran as a follow-up is then combining a couple of those different scenarios. So in these Western blots, what we've done is lapatinib treatment, which should suppress the EGFRS expression, and we've also manipulated the cells to add or remove stress to manipulate the HSF1. So this top panel up here, what it's showing is control HER2 cells treated with lapatinib with increasing concentrations. So we're blocking this receptor, we're silencing or down-regulating the EGFR, essentially. So the more drug we give, the less EGFRS expression we see. If we stress the cells at the same time, we're essentially doubly suppressing the EGFRS expression. So we're blocking this pathway up here, and we're also blocking the interest within the nucleus itself, the expression of EGFRS. We then did the reverse of that down the bottom. Again, these are control cells with increasing concentrations of lapatinib, blocking this pathway. But in these cells, we removed the stress, we knocked it down. And you can see what happens is we get a much stronger or more amplified expression of EGFRS as a result. This kind of replicates what we saw in those other cell models earlier. A couple more findings to show you. So what we did next was we created essentially drug-resistant cells. So we treated these breast cancer cells with increasing concentrations of lapatinib over the course of several weeks, up to 2 months, basically. So what we saw as we gave these cells more and more drugs, some of the cells would die, others would live on, and we continue to culture those ones that live, essentially creating resistant cells, which is what happens clinically when the drug, the cancer itself, is no longer responsive to the drug. So you can see what happens at the start. We've got very similar EGFRS expression in the control cells, and initially when we treat the cells with lapatinib, like I've shown you already, if we give some drug, we're knocking out this receptor complex, we're silencing this signaling pathway, we don't get any EGFRS expression, we get less EGFR expression downstream. But as the cells become resistant towards the end of the 2-month period, we're still treating with drugs, we're treating with much more drugs, in fact, much more than would be given clinically. We're trying to silence this pathway, but the cells no longer respond. The EGFRS is switched back on, that down-regulation process is lost. And this is what will be probably happening when a cell becomes drug-resistant, because this pathway signals for cell growth and proliferation. So that's what's happening when a cancer is not responding to treatment, it's continuing to grow and divide. The last result I'm going to show you is a knockdown. So in this final experiment, what we did was designed a knockdown cell line. Essentially, we're silencing EGR4 in different ways. So we confirmed this with a Western blot. You can see the control cells here on the left-hand side express the EGR4X, and these 2 different cell variants that we created have very little expression of this protein. And then we did a growth assay to look at how they grow over time. In the green up here, we've got quite fast growing control HER2 breast cancer cells. When we switch down the EGR4S, you can see that their growth essentially plateaus. They're not really growing much at all. And this is very similar to that result. You can remember back to 15 minutes ago when I showed you this at the start. When we were looking at these SKBR3, which is another HER2 breast cancer cell type, in the control cells, they're growing quite quickly, but when we add stress into the mix, the growth rate is reduced. Similar kind of pattern emerging when we directly influence the EGR4 itself. Okay, so to summarise all of that then, we found that HSF1 activated stress in breast cancer is linked to a more metastatic phenotype in that they're more migratory, they're more disorganised in 3D growth, but the growth rate is much slower. And what we think is happening there is that there are two possible pathways that the cancer can follow. It's either going to stay in place and grow and proliferate, or if you stress that cell, what's going to happen is it's going to push it onto a different track. It's going to shut down growth and essentially or preferentially push it into a metastatic pathway where it's going to spread out and disseminate into other areas of the body. We also found, second point there, EGR4S is overexpressed in breast cancer cells with activated HER2 pathway. That could have been through either HER2 or HER1, or a heterodimerization of both of those receptors. And hopefully I've shown you that we could see an inverse relationship that exists between cell stress and also EGR4S. So when stress is high, EGR4S is low, and then vice versa, EGR4S is up when stress is reduced, and we can manipulate that in both directions. The three key findings that we think are really interesting that has come out of this work and what we want to pursue is firstly that drug inhibition of the HER2 pathway reduces EGR4S expression. So when we use something like Metastasis, which is used clinically, this is similar to the Herceptin that I talked to you about before. All of these targeted therapies bind to and inhibit this signaling pathway. They are switching off all the downstream signaling molecules, reducing EGR4S as a result. So we think that's really interesting and potentially has clinical applications. The second key finding is that EGR4S responsiveness is lost with sustained high-dose drug treatment. So when we make the cells drug resistant, which is what happens clinically when someone's breast cancer progresses, we no longer see this inhibition of EGR4S. So we can treat it with all the target therapy we like, but nothing is happening to EGR4S. So we normally see an inhibition, but when it's drug resistant, we don't see that anymore. And the third key finding is, most of what I talked to you about today, is that cell stress reduces EGR4S expression, but is associated with greater metastatic potential. So taken together, we think that's a really exciting and interesting avenue that can lead on to some further experiments. So our next steps are to investigate the potential for EGR4S use as a biomarker for breast cancer and response to targeted therapies, because like I talked to you about at the start, these targeted therapies are really expensive, don't work for everyone. So we need a way to be able to screen more effectively and tell when or if something will work or not. It'll be beneficial for the patient and also save a lot of money for both the patient and the government as well. And also the use of targeted therapies can become even more personalised, which is great because, like I talked to you at the start, the more personalised the treatment is for the woman, the better the outcome is for the patient, and that's something we all want. So just to wrap up, I would like to acknowledge a number of people. This is a team effort, it's not just me. So the systems map, I had lots of help from Monique, Andrew and Jonathan to pull all of that together. All of that molecular biology, Chao and I worked on together really hard since 2013, so that was a lot of work. Ben and Martin from Harvard helped a lot with the bioinformatics, Jack and Chris Dow at Sunshine Hospital, and also Stuart and John Price. And the funding for all of this came from internal research grants at Victoria University, And also publication fees were covered by ProMetTre Cancer Research, which is a not-for-profit, which I helped to set up to disseminate this kind of information and to raise money for research into metastatic cancer. And just before I finish, I am doing, speaking of raising funds, the Cancer 200 Ride for Research on the weekend with my brother in honour of my mum, who died nearly 20 years ago from breast cancer. So if you would like to donate, and I'm sure all the people at Harry Perkins would really love that. And I'd like to thank you all for coming along and listening online. If you have any questions, I'll be around, so you can ask them now or come up and ask me afterwards. Also, I'm always looking for people to collaborate with on future work. So if anything has sparked an idea and you think it would be great to work on something, I'd really welcome your input, and maybe we can do some work together in the future. Thanks for your attention.
Thanks, Jeremy. That was awesome. As you said, most of it's out of my head. I did have the pleasure of reading your thesis a couple of times during your PhD, and most of the time it was just going, can you explain this in a bit more detail? Does anyone else have any questions in here? You're the only one. Oh, sorry.
Thanks, Jeremy. That was a really interesting talk. We've been looking at stress from a different perspective, so looking at headshot in the context of saving the cell in a way.
Okay.
And we're looking at it from the other perspective of immune sort of cell stress that signals to the immune system to kill the cancer cell. And I just wondered whether or not there's any relationship in the expression of these proteins with each other. So the stress molecules associated with immune recognition of the cancer cell, a transformed cell, often throws up these sort of ligands for receptors on immune cells. And I just wondered whether you looked at those molecules as well to see if there's any relationship.
We haven't looked specifically at any of the immune modulator stuff. I know that there are, speaking of heat shock or like a potential saving of the cell, not even saving, the inverse of that is actually being undertaken in some clinical trials. They're using drugs to try and counterintuitively stress the cells even more to try and push them to apoptose, but a lot of these clinical trials don't seem to have very much success, and it might be because it's not actually successfully pushing them into cell death, and it's in that nasty area where it's just sufficient to make them somewhat more aggressive. But sorry, that didn't really answer your question at all.
That's fine. We're using tools like CRISPR, etc., to make them more immunologically visible.
Yeah, okay.
Rather than thinking about it from a heat shock context, so it's sort of the other way. But I just wondered whether or not similar tools might be useful in this instance.
Yeah, potentially.
To modify the expression.
Yeah, potentially, yes.
Great talk, Jeremy. So do you think then that the EGFRs might be a suppressor of the epithelial to mesenchymal transition to help mobilise those cells into that metastatic?
Potentially, yes, because there's research that shows that switching on stress, heat shock factor 1, is implicated in that whole process of epithelial to mesenchymal transition, because EGFRs is a transcription factor, so it's a downstream regulator. It will bind to the DNA and trigger a whole suite of genes to be ramped up. That could be potentially one outcome. We haven't looked at it specifically in our work, but it's a possibility that we can look at in the future. Yeah.
And what's missing in that exon 1 deletion? But the small, what does that?
Well, all of the DNA, there's 3 DNA binding domains, and all of them are located specifically in the exon 2 region, which is why the X version is specifically a splice variant derived from exon 2. So we have hypothesizing we haven't done any direct interaction experiments, but it should still be able to bind to the DNA, still be able to elicit that response. The full-length EGFR is transiently expressed in a lot of immortal cells. So it was first discovered in the developing brain when and neural cells can live across your entire life. It's also localized in the stem cells in sperm. So that full length is important, it seems to be, for lifespan, ongoing differentiation and proliferation, and this S stress shorter variant seems to be related to growth, obviously, because it's downstream of the HER2 pathway, but then potentially metastasis as well.
Anybody else?
I don't know much about all this stuff, but if you look at, I suppose, the response to stress, you know, it can be a whole bunch of different things, oxidative stress or, as you mentioned. Ultimately, if you bring that back to stress on the individual that's got the breast cancer, a lot of the alternative remedies, I suppose, for cancer are based on reducing stress. Things like yoga and mindfulness and Reiki and all those things. They should be reducing stress. So then, if the paradigm is that cellular stress that causes the metastasis and everything else, why don't all those alternative therapies work?
Well, there's a number of things I could say about that. So, firstly, everyone is unique. Everyone is different. Everyone's cancer is different. Everyone responds to stress differently. So what stresses me might not stress you, and vice versa. It's a lot of the clinical trials. So there have been randomized controlled trials. I can recommend a really good paper that was published earlier this year, which is published in Nature, to review this entire topic. People's life history, if we're talking about psychological stress, like there's so many factors at play in how an individual will respond to an event that would be stressful or not. And people will use different techniques to moderate that stress as well. So what might, like yoga might work for you, might not work for you. So it's a complicated area, I think, to unpick. I hope that answers your question.
Yeah, well, I suppose, you know, you can do yoga for so many hours a day, but then you've got to live for the rest of it. So.
I'll just follow up on that because you focused on financial stress and that would be contemporaneous with the cancer. So given the different health systems in Australia and in the US, you would expect that the success rate in the US would be substantially lower if financial is a, is that the case?
Potentially, I am not aware of any research that has looked at contrasting health systems and how that plays into the outcomes for the patient. So yeah, that's potentially something that might be interesting to have a look at. Yeah, Herceptin has, the patent for that has just recently expired, so it's no longer $50,000 a year or $100,000 in the US. But there are other drugs in that class. So there's patritumab, which again is government subsidised for hundreds of millions of dollars a year. So they are expensive drugs.
It would seem you'd be able to look at that because the way the insurance system, et cetera, works in the US, you can get a whole array, a whole distribution of financial stressors on the patient at the moment.
Okay.
So,
No, it's fine.
oh,
This might be the million or billion dollar question, but in your drug resistance induction, do you have any idea where that's working, operating at? Is it at the drug receptor interaction, or is it a specific pathway downstream of that?
Well, heat shock factor 1, the heat stress response is implicated in drug resistance. So as a cell becomes more aggressive, more drug resistant, the heat shock factor will be upregulated. We're not really sure from what we've done what's actually happening within the machinery of the cell. We haven't gone into that level of intricate detail, but I would expect that there's something that's gone right there, because obviously it's then become more aggressive, but it's also switched on the growth pathway as well, because that's what's happening if the EGFR is upregulated. So I can't answer that question with any clarity, but that's what we think might be happening. Yeah, something to look at.
yeah, one more.
I'm trying to formulate one, but
Maybe you can talk about it as well.
Yeah.
Well, then, thank you very much for being here this afternoon. Thanks, Jeremy, for coming and sharing the research with us. And for all of the questions. As Jeremy said, if you've got some other questions, go have a chat to him. He doesn't mind having a talk. Enjoy your afternoon.
Thanks, everyone.